morning student, we will continue topic protein metabolism computation number 5.4 and today's subtopic is metabolism of branched chain amino acid and their associated disorders. In our body we had three branched chain amino acid, that is valine, leucine, isoleucine. Valine is glucogenic amino acid, that means on catabolism of valine glucose is formed. Leucine is purely ketogenic amino acid, that means on catabolism of leucine ketone bodies are synthesized. Isoleucine is both ketogenic and glucogenic. So these are three branch chain amino acids and all are essential. These amino acids serves as an alternative source of fuel for brain, especially under starvation. All these three amino acids undergo similar sequence of reaction during their catabolism. Important enzymes of catabolism of branch chain amino acids are three. First, transaminase, second, alpha keto acid dehydrogenase, and third is FAD dependent dehydrogenase. So these are the three enzymes of first three steps of catabolism of all these three amino acids. In the catabolic pathway, the second step requires enzyme alpha keto acid dehydrogenase. So this is not a enzyme, but it is a enzyme complex. It consists of 3 enzymes and 5 coenzymes. And these coenzymes are thymine pyrophosphate, coenzyme A, lipoate, NAD+, and FAD. If this enzyme is deficient, then it will lead to a disease called as maple syrup urine disease. So this is the catabolic pathway of all these three branch chain amino acids. First 3 steps are same in all these 3 amino acids. If you see, valine is converted into alpha keto isovalerate. Here transamination takes place, amino group is removed, which is accepted by alpha ketoglutarate to form glutamate. The coenzyme here is pyridoxal phosphate. The same happens in the first step of leucine, where leucine on transamination forms alpha keto isocaproate. Products are different because number of carbon atoms present in these amino acids are different. Similarly, isoleucine on transamination forms alpha keto beta methyl valerate. Next step is oxidation. Here oxidation takes place by removal of 2 hydrogen atoms along with simultaneous decarboxylation. So you can also name it as a oxidative decarboxylation steps. So it occurs in all these 3 amino acids. In the first case, alpha keto isovalerate on oxidative decarboxylation forms isobutyryl CoA, coenzyme A is added. Similarly, alpha keto isocaproate is converted to isovaleryl CoA, and alpha keto beta methyl valerate is converted into alpha methyl butyryl CoA. The enzyme for all these 3 steps is same, and this enzyme is second enzyme alpha keto acid dehydrogenase. Third step is also same, where in case of valine catabolism, isobutyryl CoA is converted into methyl acrylyl CoA. Here again one more oxidation takes place by removal of hydrogen atoms, but here hydrogen acceptor is FAD. In the previous step, the hydrogen acceptor is NAD. In case of leucine, isovaleryl CoA is converted into beta methyl crotonyl CoA, again by oxidation. In case of isoleucine, alpha methyl butyryl CoA is converted into tiglyl CoA, again by oxidation. So here all these 3 steps require a enzyme called as FAD dependent dehydrogenase or simply dehydrogenase. Now the fourth step, from fourth step the catabolism of this amino acid differs. Methyl acrylyl CoA undergoes hydrolysis to form beta hydroxy isobutyrate. Again here two hydrogen atoms are removed which are accepted by NAD. Later on it is converted into malonyl CoA. Malonyl CoA is converted into succinyl CoA. Here coenzyme B12 or cobalamin is required, and then succinyl CoA enters into TCA cycle where it is converted into oxaloacetate first, and later on that oxaloacetate is converted into glucose by gluconeogenesis pathway. Therefore, valine is a glucogenic amino acid. In case of leucine, in the fourth step, carboxylation takes place. Beta-methyl crotonyl CoA is converted into beta-methyl glutaconyl CoA. Just like other carboxylation reactions, it requires coenzyme biotin. Then hydrolysis occurs, and in the next step, beta-methyl glutaconyl CoA is broken down into two compounds, that is HMG CoA and acetyl CoA. The HMG CoA means beta hydroxy beta methyl glutaryl CoA, and this HMG CoA in the next step is converted into acetoacetate. So acetoacetate and is a ketone body, and this acetyl CoA is converted into ketone bodies. So that means the product which are formed by breakdown of HMG CoA, that is acetoacetate and acetyl CoA, both are converted into ketone bodies. So therefore leucine is a ketogenic amino acid. In case of isoleucine, in the fourth step, tiglyl CoA undergoes hydrolysis to form beta-methyl beta-hydroxybutyryl CoA, which undergoes further oxidation in the next step to form methyl alpha methyl acetoacetyl CoA, which is broken down into propionyl CoA and acetyl CoA. Propionyl CoA is converted first into succinyl CoA, and this succinyl CoA is then entered into the TCA cycle where it forms oxaloacetate, and this oxaloacetate through gluconeogenesis forms glucose. The acetyl CoA which is formed as a second product is converted into ketone bodies. Therefore isoleucine is both glucogenic and ketogenic amino acid. Now coming to the inborn errors in the catabolism of branch chain amino acid, the most important error is maple syrup urine disease. It is also called as branched chain ketonuria because branched chain amino acids are not catabolized in the body, therefore they get excreted into the urine. Incidence of this disease is 1 per 1 lakh births. The name originates from characteristic smell of urine, which is like burnt sugar or maple sugar due to excretion of branched chain keto acids in the urine. The basic biochemical defect in this disease is deficiency of enzyme alpha keto acid dehydrogenase, which catalyzes the second step of these branch chain amino acids. Clinical findings include this disease starts in the first week of life. It is characterized by convulsions, severe mental retardation, vomitings, acidosis, coma, and death may occur within first few years of life. Coming to the laboratory diagnosis of this disease, if you analyze urine, you can find the presence of branch chain keto acids like leucine, isoleucine, and valine in the urine. Here Rothera's test is also positive, just like ketonuria, but the difference here is if you boil the urine and then perform Rothera's test, in case of ketonuria the Rothera's test is negative after boiling, but here even after boiling you will get Rothera's test positive. The final diagnosis or confirmatory diagnosis is done by analyzing the deficient enzyme, which can be done prior to 1 week after birth. Treatment of this disease include giving a diet low in branch chain amino acids. Sometimes mild variant of this disease is seen, which is called intermittent branch chain amino acid ketonuria, and this will respond to high doses of thiamine. So what is the role of thiamine here? I have told you that in case of maple syrup urine disease, the deficient enzyme is alpha keto acid dehydrogenase. It is not a enzyme, but it is an enzyme complex which requires 5 coenzymes: thymine pyrophosphate, coenzyme A, lipoate, NAD+, and FAD. This thymine pyrophosphate is obtained from thymine. So if you give thymine in high doses to a patient with mild variant of maple syrup urine disease, then this thymine stimulates this enzyme and activity of enzyme is increased. Therefore, the symptoms may be lowered. The final treatment of disease is liver transplantation, which has been successfully tried in some cases of maple syrup urine disease. One more inborn error is seen, which is called as isovaleric aciduria. This disease is affected due to deficiency of third enzyme, that is FAD dependent dehydrogenase, but it does not affect all these 3 amino acids. It only affects the catabolism of leucine. So these are the first 3 enzymes that I have told you. The third enzyme is dehydrogenase or FAD dependent dehydrogenase. In this disease, only the catabolism of leucine is affected, where enzyme is isovalerate dehydrogenase. It is shown here. This is the leucine catabolism, and the third step, the dehydrogenase enzyme, FAD dependent dehydrogenase, or complete name is isovalerate dehydrogenase enzyme, is deficient. The catabolism of other amino acid is normal in this condition. So other 2 amino acid catabolism is normal. Clinical features includes severe metabolic acidosis, neurological deficits, very high amount of abnormal metabolites are excreted in the urine. The characteristic offensive odor of urine is the first sign of abnormal excretion of these metabolites, and this is also fatal in early childhood. But this disease is very rare as compared to maple syrup urine disease. So this is about the catabolism of branched chain amino acids and its associated disorders. Thank you very much.